Okay, right. We're all good to go. So welcome everyone to MND Research Australia's first edition of State of Play. This seminar series is to replace our usual MND Connect session. Obviously, at the moment, it's a bit difficult to get everyone together. So hopefully this gives us a fantastic forum to bring the research to you, into your living rooms, into your offices instead. Across the series, we'll be showcasing some of our top researchers and their projects right across the MND research spectrum, from basic mechanisms that cause MND right through to better models of caring for our patients. So tonight, we have two researchers talking about different ways we can model the MND disease process in the labs. First up, we've got Samantha Barton from the Florey Institute in Melbourne, talking about her work on stem cells. And then second, we've got Adam Walker from the University of Queensland telling us about his work in mouse models. Right, throughout the seminars, please feel free to ask questions using either the Q&A button at the bottom of your Zoom screen, or if you're on Facebook, you can use the I think you can use the chat function in Facebook or the comment function to send us questions through there, and we'll be picking those up. We'll have the two, both Adam and Samantha will be answering questions at the end of the talks together. So we won't have questions directly after each talk. There'll be a joint question and answer session at the end of the two talks. So I think that's enough from me. And now let's move to the main event, and we have Samantha up first telling us about stem cells.
Thanks, Gethin. Let me just share my slides. Okay, can you see those okay?
Yep, we can see those.
Okay, great. All right, well, thank you for the introduction. My name is Sam, and I'm a postdoctoral researcher working at the Florey Institute of Neuroscience and Mental Health. Today I'm going to be giving you quite a broad talk on the uses of stem cells in research for motor neuron disease. So I'll begin with quite a broad introduction that will introduce both me and part of Adam's talk as well. So here we have a schematic depicting a cross-section of relatively near to the front of the brain, and we have the motor cortex up here in both hemispheres. And you can see our motor nerves begin in this motor cortex. We have our cell body, these little black dots. Then they extend their axons down through the back of the brain, out through the pons, down the corticospinal tracts into the spinal cord, and down here till we're probably about 3/4 of the way down the spine, where they synapse with these next nerves called the lower motor neurons. So these lower motor nerves, their cell bodies begin in the spinal cord, and they project their axons out of the spinal cord towards the skeletal muscle. And so this whole process is involved in everything that we do that controls movement. So me talking to you right now uses this same pathway where our message begins in the brain and passes all the way down to the skeletal muscle. So what we know is that in patients that have MND, this whole pathway is disrupted. So they have dysfunction in these nerves, and this leads to eventual death of both these upper motor neurons as well as these lower motor neurons. And the loss of these neurons means that we don't have innervation of the muscle. And we know that when muscle doesn't receive activity, it atrophies and dies. And so this leads to the more hallmark symptoms that we're familiar with of muscle weakness and paralysis that can then lead to issues with walking, general moving, and then issues with swallowing and breathing. So in order to understand these processes, or our jobs in the lab is to try to work out what is going wrong in these nerves that's leading them to dysfunction and die. And so we have a few model systems that we use in the lab to be able to understand this. The first one is imaging, which you're all probably familiar with, things like MRI and DTI imaging. And so this can be done non-invasively of patients throughout their disease course. And so we can take these images both from at the very onset of their disease, as the disease progresses, and then towards the end of disease. And we can look at the gross changes that are happening in their brain, such as in this image here. So this is obviously a strength. It's non-invasive, and it's giving us real-time images of a brain that's undergoing MND. But the downside of this is that we can only see the gross brain. We can't actually see the cellular changes that are occurring like these individual motor neurons. So we can then add a complementary model system, and another example is using human postmortem tissue. And so this is when people with MND generously donate their brains or spinal cords after they die. And this allows us to look at this tissue and look at the cell types specifically. And so it's complementary to the imaging because it means that we can look at a much higher resolution and try to more mechanistically look at what's happening in the brain that's leading to these neurons dying. So again, this is a very strong model system, but a limitation of this is that obviously postmortem samples are taken when the patient has succumbed to the disease. So what we're looking at is the very worst case where these cells, the majority of them have already died. So again, we can look at some of the pathways that are involved. We can't look at them at what's happening during the disease course. And so it's probably here that I should mention something that's very obvious for something that we kind of forget, is that the brain is extremely inaccessible. If you think about any other disease, such as even cancer is a very good one. If someone has cancer, we can very easily biopsy it and look at their specific cancer that they have and exactly what's happening within that cancer. Whereas someone with a neurological disease, we're very limited in our capacity to actually see what's going on in that precise moment in their brain. So this has led to the introduction of a couple of other model systems, the next one being animal models, which Adam will go into much more detail talking about after me. But the brilliance of animal models is that we can model aspects of disease and so therefore start looking at these animals preceding disease onset, during disease, and also then at end stage. So we can kind of get a snapshot of what's happening throughout the disease course, which is imperative in order for us to understand what's happening and therefore identify things that we can treat and translate to benefit to patients. The final model system, and the one that I will talk about today, is human stem cells. And so I will now crack on with giving you an overview of what a stem cell is. So in order to explain what a stem cell is, probably the best example to give you is the most common one, which is the embryo, the developing embryo. So we know that when a sperm fertilizes an egg, it leads to the generation of a cell, which continues to divide and give rise to the developing embryo, the fetus, and then us. And so we know that in this early embryo stage, we have this bunch of cells that are called master stem cells, which means that they can become any of the cell types of the human body. And so the brilliance of this is that this stem cell has the capacity to become anything. It can become a heart cell, a brain cell, a skin cell. But once the stem cell decides what its fate is, it can't go backwards to becoming a stem cell. This has led to some research into using fetal stem cells because it allows us to look at the generation of something such as the brain, but it doesn't really help us in modeling brain diseases, given that we know that once the stem cell decides what it wants to be, such as a brain cell like a neuron, we can't then go back to the stem cell. So this doesn't really help us combat the issue of the inaccessibility of the brain. This was until this dude came along. His name is Shinya Yamanaka, and he's a Japanese neurologist and scientist who in 2012 won the Nobel Prize in Physiology or Medicine for discovering that he could actually turn one of these terminal cells back into a stem cell. And so what he did was that he took an adult human patient and he took a biopsy of skin cells, so the epithelial cells here. So they're an end stage cell which can't normally become a stem cell. He cultured them in a dish and then added a cocktail of factors which genetically manipulated these cells and allowed them to actually reprogram into becoming this master stem cell. So you can understand why he won the Nobel Prize for this. These cells are called induced pluripotent stem cells. So induced because we've forced them backwards against what they normally do. Pluripotent, they have many potentials, and then stem cells, they have the capacity to become any stem cell, any cell type. So this means that because of Shinya Yamanaka, we now have the capacity to take adult stem cells, so we avoid the ethics of using fetal stem cells. And so this gives us the capacity to now grow cells like the brain cells in a dish and finally allow us to interrogate the biology of the brain and the spinal cord and combat that kind of inaccessibility that we've been challenged with for so long. So this is what the cells look like growing in the dish. These are our induced pluripotent stem cells. Each of these colonies is probably a few thousand cells that grow really happily and compactly in these little colonies. What we can then do is keeping in mind that these stem cells are taken from an adult patient, so they're genetically identical to the person that we've taken them from. We can then add another cocktail of factors to genetically program these to mimic the development of what happens in brain development in a fetus. So in essence, we're telling these cells they want to become brain cells, but in a dish. So they start expressing some of these factors and start forming the morphology of what these cells look like in the brain and the spinal cord. And what this means is that we can start generating some of the cells of the brain. So if you think back to that first schematic, this is the motor nerve in the yellow here, the cell body projecting its axon down and synapsing with the next nerve. But what we know is the brain and spinal cord is made up of much more than just these nerves. They're surrounded by a much more complex population of cells called glia, such as the microglia, which is the immune cell of the brain, the oligodendrocyte, which is the myelinating cell of the brain, and then the astrocyte, which is kind of the glue that holds everything together. And then, of course, we have the vasculature, which provides all the blood and the oxygen and the metabolic factors that we need in the brain. So the brilliance of our system is that we can start with these pluripotent stem cells that are genetically identical to our original patient donor. And we can, in effect, in a dish, make what their motor neurons would look like within their brain and spinal cord. And we can also make these other cell types, such as the astrocyte, the oligodendrocyte, and the microglia. So in theory, we are growing these brain and spinal cord cells in a dish that mirror that initial patient. And the next level from this is that while we can look at these cells in isolation and work out how are these cells different to a cell from someone that doesn't have MND, but we can also grow them in a much more complex three-dimensional structure and work out how these cells talk to one another and what's happening at a more sophisticated level. So now that I've kind of given you the introduction to iPSCs, or induced pluripotent stem cells, I'm going to discuss 3 ways that we can use these stem cells in our research. The first way is to investigate some of the causes of MND. The second is to use them to find a treatment for MND. And the third is how we can use the stem cells as a treatment for MND. So firstly, understanding the cause of MND. So I'm just going to give you a brief overview of some of the experiments going on, and this is actually my own research from the lab. So we start with our person living with MND, and we take our skin sample, as I've discussed. We grow the skin sample and we re-derive these induced pluripotent stem cells. And then we take the same cells, but from a control patient. So in effect, we're growing these stem cells from someone with MND and someone without MND, and we can compare and contrast between them. So my particular interest is actually on this cell type called the oligodendrocyte. So it's the blue cell in this schematic. And they have the very critical function of producing what's called myelin, which is this fatty substance that goes around motor neurons. So it functions much like if you think about your kettle and you've got your power cord that you plug into the wall. It's like the rubber on the outside of the wire, so that it makes sure that the conduction is maintained and that you have electricity to your kettle. So the oligodendrocyte functions much like this, but the added bonus is that it can also shuttle metabolic and trophic factors through this myelin into the neuron. So it's critically important in order for the neuron to function normally and happily. So my interest is in working out whether this oligodendrocyte is the same or whether it has its own dysfunction in ALS. So like I said, we have our stem cells from our MND patient and from our control patient. And then we can grow our oligodendrocytes from our MND patient and our control patient and compare and contrast if they're different and how they're different. And so one of the ways we can do this is we can measure how much fuel these oligodendrocytes produce. Keeping in mind that oligodendrocytes supply this fuel to these nerves, we can measure whether oligodendrocytes in MND patients can provide the same level of fuel as those from a control patient. So here in this graph here, we have 2 MND patients, and you can see that the capacity to produce fuel is at about 0.17. If we compare this to 2 control patients, you can see that they have almost a 2-fold increase in the amount of fuel that they can produce. And so what this means is that indeed in ALS, oligodendrocytes are not producing the same amount of fuel in order to support motor neuron function, and therefore could be contributing to the disease. And so this just highlights the way in which we can use these stem cells to work out what pathways are affected. And by identifying these pathways, we can then target them with drugs to try and rescue these pathways, and therefore potentially continue the support of these motor neurons and prevent their dysfunction or death. So I'll now move on to the second way we can use stem cells, and this is to find a treatment for MND. So again, we start with our induced pluripotent stem cells from our MND patient, but this time we're going to make them into the motor neuron, which is the yellow cell here, the one that we know undergoes dysfunction and death in people living with MND. So this is a little video here showing our stem cells here becoming motor neurons. So you can see that over time they're changing their morphology. They're starting to form their little cell bodies, and then they're projecting their axons out, much like that schematic that I showed you before. So then by the end, we have these mature motor neurons. And what we can do is we can plate them into these tissue culture plates that have many, many wells. So they can have 96 wells or 384 wells. And so it means that all of these wells become filled with these motor neurons. And the brilliance of this is then we can screen drugs on these motor neurons. So they can either be motor neurons generated from someone with MND or from a control patient. And then we can add the drugs to try and manipulate this process to see what pathways are important and how we can sort of rescue any deficit identified in these motor neurons. And so the strength of this is that it can be a highly automated process. So at the Florey, we have a brilliant setup where this is all done robotically and at really high throughput. So the robot here is grabbed the 96 well plate, and all the motor neurons are already growing in this plate. And so the robot can then add the drugs to all the wells, and this can be done really quickly, allowing us to screen tens of thousands of drugs all at once. So this can be done on many different motor neurons from many different patients, which really allows us to in-depth look at the capacity of drugs. And so we're very fortunate to receive funding from the Fight MND to allow us to do this. And so stay tuned because it's a really exciting endeavor. Oh, might not play it again. Right, so the final thing that we can use induced pluripotent stem cells for is actually a treatment in of themselves. And because this is kind of a more recent advancement, the example I'll talk about is actually from Parkinson's disease, and then I'll bring it back to motor neuron disease. So back in the 70s, some research took place whereby scientists took fetal tissue and they isolated the part of the brain that gives rise to the cells that are affected in Parkinson's disease. These are called dopaminergic neurons. So they're the motor neuron equivalent in Parkinson's disease. So they isolated these cells and then they transplanted them directly into the brains of people with Parkinson's disease, with the theory being that by taking these fetal dopaminergic neurons, they would replace the ones that have been lost in these patients with Parkinson's disease. And then obviously these patients needed to be immunosuppressed so they wouldn't reject the grafts. So this graph here shows some of the data that came out of this experiment. So this baseline here was untreated patients. So you can see that their motor activity didn't change and actually declined a bit towards the end. Whereas there are 3 experimental groups that each contained a subset of patients that received these transplants. So you can see in these 2 here, there was a significant decline, but decline is good in this case. So this decrease actually indicated that after receiving these transplants, these patients had improved motor capacity and sort of decreased symptoms. And then you can see in this 3rd group here that they did have an increase in their motor function. However, it did then decrease and return back to baseline. So whilst there were still some obvious sticking points, the first one being the ethics of using fetal tissue for transplants, there was also then some discrepancy in why some patients returned to normal and some didn't. But it really raised the question of whether we could replace the cells that are being lost in brains and spinal cords of patients, and whether this is an ideal avenue that we can go down. So this has obviously progressed since the 70s, and the current research is now starting to incorporate in induced pluripotent stem cells. The benefit of this is that in this schematic, again, we can take the biopsy of the skin cells and grow them here, reprogram them into becoming stem cells, then make lots of stem cells, and then we can differentiate these into our dopaminergic neurons in the case of Parkinson's disease. So this is much like the process I've already discussed. And then we can transplant these into the patient with Parkinson's. And so this is obviously not happening yet, is the, I guess, what people are aiming towards. But the brilliance of this technique is that the original cell comes from the patient themselves. So in theory, they won't need any immunosuppression in order to receive this. But I want to highlight that this is a very complicated technique with many questions still unanswered. So it is still a while off, I think, until this will be optimized. Questions such as, will the transplanted cells replace the diseased or the dead cells? Will the transplanted cells only support the survival of the existing cells? Will the transplanted cells physically support the cells, or will they secrete factors that chemically support the cells? Does it matter what type of cell or how many cells we transplant? Should the transplanted cells come from the same person, or can they come from a different person? And can we genetically alter the cells prior to transplantation to express certain factors or proteins? And then I guess the fundamental question of whether we need the whole cell at all, or whether we can kind of identify what part of the transplanted cell that is doing the reparative work, and whether we can just mimic that instead of transplanting whole cells. So now I'll bring it back to the context of MND, because obviously this is a hot topic and there are some clinical trials that have begun. I want to highlight that none of them are on induced pluripotent stem cells yet, but I'll work through a couple of the ones that have gone so far. So the first one, and the one that's been most recently in the news, is using mesenchymal stem cells. So if you think back to my first slide where I had the stem cell becoming the terminal cell types, mesenchymal stem cells are a few steps down in the lineage. So they're not quite master stem cells, but these type of stem cells can be isolated from the bone marrow. And so they still have some potential to become various cells such as muscle cells and blood cells and those kinds of things. So this trial of transplanting mesenchymal stem cells into patients with MND underwent phase 1 and phase 2 and was shown to be safe and beneficial in a subset of patients. The phase 3 trial was recently completed. They released a statement last week, and they showed that there was no significant effect between groups, but they did say that there was potentially some benefit in subsets of patients. Another clinical trial going on is using neural stem cells. So again, we're not quite at the master stem cell. We're a little bit down the lineage where they've already decided they want to become brain cells. And so like I said, due to the inaccessibility of the brain, these can only be isolated from fetuses. This underwent phase 1 safety trial and was found to be safe, albeit in very small numbers of patients. And so the phase 2 is currently underway. The third trial was using glial restricted progenitor cells and neural progenitor cells. So again, the master stem cell has already chosen it wants to be a brain cell, so we're a little bit further down the lineage, and again, can only be taken from fetal samples. And again, the phase 1 safety trial is underway. With regard to induced pluripotent stem cells, these have not made it to human trials yet, in MND anyway, but certainly they're starting to undergo optimization in animal models, and some of this work is even being done at the Florey Institute, so stay tuned. This is another really exciting avenue. So finally, I'd just like to wrap up, and I'd just like to acknowledge all of my lab members. This has been how our lab has met this year during COVID. This is us last week in our MND t-shirts celebrating the walk to defeat MND with all our favourite walks in the background. So thank you. All right, so I would now like to pass on to Dr. Adam Walker, who will now talk to us more specifically about the use of mouse models in medical research.
Great, thanks very much, Samantha. Can everyone see my screen here?
Yep, all good, Adam.
Okay, great. So I'm going to be speaking today about what mice can teach us about motor neuron disease, following on from Samantha's really great introduction there. So there are two main reasons that we use mice in motor neuron disease research. The first of these is to study disease mechanisms. So understanding what's going on in motor neuron disease. So using mice, we can look at proteins, cells, nerves, whole brains, and also spinal cords in mice that we have modified to have MND pathology. So shown here's a diagram of a mouse, and you can see the brain and the spinal cord of the animal. We can take the brains and spinal cords from these animals. If we look at a cross-section of the brain, for example, shown here, we can find the specific area of the cortex or in the brain of these animals, which we know contain the upper motor neurons, which are involved in motor neuron disease. And likewise, if we take the spinal cord and look at a cross-section of the spinal cord, we can find the area in which the lower motor neurons reside within the spinal cord. This is the ventral horn shown here. So the other thing that we can use mice for in MND research is to test drugs. So preclinical trials in which we can see if drugs are effective before we go into people. The questions here are whether we can prevent disease and whether we can treat disease. So a little bit of a history of mouse models of motor neuron disease. We've been using mice for decades in studies for motor neuron disease. It really kicked off in the early 90s. The first inherited causes of motor neuron disease were first reported in 1993, and these were mutations in the gene SOD1, which many of you will know about. So a year after that initial genetic study discovery was made, the first genetically modified MND model mice were produced, and these have expression of a mutant form of SOD1. These mice have a remarkably consistent MND-like disease, which presents as gradual weakness leading paralysis requiring euthanasia. So one way that we can see these mice is mice really like to grab hold and pull things, and so we can test the function of their muscles in a grip strength test. So that's shown down the bottom here. So if we look at a normal mouse over time, as time progresses, they're able to pull this bar at the same rate over time. However, these mutant SOD1 mice are decreasing their ability to produce force to pull on a bar like this over time. And this is a really good indicator of disease progression in a mouse model. Of course, it's really important to consider ethics in terms of performing animal models in diseases such as motor neuron disease. Clearly, the mice are really well taken care of, and we have to take into account the benefits of the outcomes of our research for people living with motor neuron disease, as well as ensuring that we're taking care of the animals throughout these studies as well. So what are the main advantages of using mice in MND research? So mice are mammals, and so they have nerves and muscles, which are quite similar to humans. But they are small, they're easy to care for, and they're also easy to monitor movement changes using, for example, the grip strength monitor that I showed there. One of the advantages that Samantha touched on is that we can also undertake studies in which we look at the early changes in disease. So we can't take a brain or spinal cord from people who have disease, but we can do that from mice. And this really allows us to look even before symptoms have started in animals, what are the very early changes that are occurring that drive motor neuron disease. It also allows us to study interactions of different cell types within the body. So neurons, glial cells, muscle, and inflammatory cells within a whole animal that's undergoing disease. And it also, we can use MND mice to test drugs. As with anything in science, there are disadvantages, of course, of using mice in MND research as well. So we need to consider that mice are not simply just little humans, that they actually are very different to humans, and we need to structure the questions that we're asking scientifically in order to make sure that we're able to get out the answers that we need. So mice have a short lifespan. They only live for a couple of years, which means that in studying disease such as motor neuron disease, that takes quite a long time to progress. We may miss some of those features in a short-lived animal like a mouse. The nervous system is quite complex, but it's less complex than humans, of course. The mice that we use are not very genetically diverse, which is quite different to humans, which are very diverse in our backgrounds. So far in studies of motor neuron disease in mice, unfortunately, there's been little success in actually translating drugs that have been studied in mice into people. And we need to keep in mind that the goals of our research are not to cure mice, but we want to cure people. So we need to make sure that the questions we're asking are right and that the models we're using are correct as well. It's important to realize that the mutant SOD1 mice that are used in lots of studies and have been really important in our understanding of motor neuron disease processes, they don't actually develop the typical MND pathology that's seen in almost all people who have motor neuron disease. So the question is, how do we understand motor neuron disease pathology, and can we make better MND mice? So understanding MND pathology is important so that we can progress in the research. So we now know about 15 years ago, a protein called TDP43 was found to be the main pathological protein in over 97% of cases of MND. So TDP43 is an essential protein which is found in every cell in the body, but in motor neuron disease, it clumps together and forms a pathology which is remarkably distinct from how it's normally found. So I like to think of a neuron like a bowl of muesli. So TDP43 is the oats. The oats are essential within this bowl of muesli. If you get rid of the oats, then you don't have any muesli anymore. The neuron can't survive. But just like a bowl of muesli, if you forget about it and you don't do the washing up, then the oats will clump together and form these really hard clumps within the bowl. And this is what happens with TDP43 in motor neuron disease. For some reason, which we don't know why, TDP43 clumps together and forms this pathology, which is found in the neurons in the brain and the spinal cord. So if we look at a slice of someone who's passed away from motor neuron disease, we can see that TDP43 normally interacts with the DNA, and in some neurons we can find these really large clumps or inclusions of TDP43 protein, which cause the neurons to degenerate. So in making MND mice, we set out to create a mouse model that has pathology that is reminiscent of this TDP43 pathology found in most people who have motor neuron disease. So we genetically modified a strain of mice to allow us to produce these abnormal clumps of TDP43 in the brain and the spinal cord. If we look at a cross-section of the spinal cord from these animals and we perform staining to look at this TDP43 clumping, we can see in the large motor neurons we have these TDP43 clumps in the mice, which is very similar to what we see in people as well. If we look at muscle in these mice that have this TDP43 pathology, we're looking here at the Tibialis anterior muscle, which in humans runs along the front of the leg. In a normal mouse, this muscle has these nice round plump muscle fibers. And in our TDP43 mouse model, we see that these fibers become shrunken and the mice gradually over time lose muscle mass, which is reminiscent of what we see in human disease as well. So if you look at the phenotype, the behavior of these animals that have this TDP43 pathology, we see that they have progressive movement problems. So a normal mouse, if you hold it up by its tail, will spread its hind limbs. It's a normal reflex. And in the TDP43 mice, we see that they hold their hind limbs quite close together. And so this is a really key feature of neurodegeneration in an animal. Another way that we can test motor movement in a mouse is to use a rotorod. This is a rotating wheel in which we put the mouse on and time how long they can run on this wheel before they gently fall to the platform beneath. So what we see over time is that normal mice can keep running. They love to run. And so over time, they'll be able to perform this task for many, many, many weeks and months. However, in the mice that we've engineered to have this TDP43 pathology, over time, they lose this ability to perform this running task to the point where they've lost weight and showing paralysis and require euthanasia. So the question that we have from using these models, and now that we have a model that has both this TDP43 pathology, which is similar to human disease, and also have this progressive phenotype, is what is actually causing the progression of the disease. So in order to do this, we've performed studies in which we look at the brains and spinal cords of these animals and use fluorescent dyes and microscopy, in which we can look at what proteins are associating with TDP43. So just like a bowl of muesli, TDP43, the oats, when they dry out and clump together, they're not just there by themselves. If you have fruit sultanas in your muesli, then that will also clump together with the oats when it dries out. And it's similar to what happens here in the pathology with TDP43 protein. When you have the TDP43 clumping together and forming these inclusions, this pathology within the neurons, that they pull other problem proteins into this pathology as well. And so these proteins that are involved in this pathology may be having other functions in disease, which is driving the progression as well. So the questions that we have now moving forward with this research is, what are these other proteins doing on disease and how are they involved in causing the neurons to die? So one of the questions that we can use these mice to answer is, what happens if we get rid of this TDP43 pathology? So we know that in people who have passed away with motor neuron disease, they have this TDP43 pathology in the neurons, in the brain and the spinal cord. In our mouse model, they have this pathology as well, but we've engineered them to have an artificial genetic system in which we can control the buildup of this pathology. So we use these mice and we allowed them to form the pathology in the brain and the spinal cord, and then we use this artificial system to turn off the pathology. So what we saw after a period of time is that the brains and the spinal cords of these mice no longer had this pathology after we stopped it from being produced. So this means that the nerves in the brain and the spinal cord are able to get rid of the TDP43 pathology if they're given the chance to do that. The other remarkable thing was that when we looked at the behavior of these animals, once we stopped this progression of the disease by stopping the accumulation of this pathology, the mice actually recovered. So if we look at the running ability of these mice, you can see that over a period of 6 weeks, normally they lose the ability to perform this function. If we stop the continued accumulation of this TDP43 pathology in the neurons, over a couple of weeks, the mice regain their running ability and they're able to perform almost at the same level as a normal animal. So this indicates that over time, the mice, if we're able to prevent this TDP pathology from accumulating, can actually improve. So if we look at the survival of these animals as well, as I mentioned, usually over time, these mice require euthanasia because they progress to the point where they have almost complete paralysis. However, in the mice that we allowed the pathology to accumulate, but then stopped it and allowed the pathology to be cleared, these mice lived months and months and months longer than we would expect. So this indicates that even after the disease has started, even after we have TDP43 pathology, if we can find ways in people to get rid of this TDP43 pathology, then that may be beneficial and allow for recovery in function if there are some remaining motor neurons that are functional. So the other way that we can use these mice is as a preclinical tool, so testing drugs. So the one drug that's been approved in Australia for treatment of motor neuron disease is riluzole. The first phase 3 clinical trial of this drug was way back in the early 90s. The mechanism of this drug is still not really very well understood, but we do know that it can give a couple of months extension of life for people with MND, or possibly longer in some people. We do know, however, that this drug doesn't work in those SOD1 mice that have been used for a very long time as well. But we know that the SOD1 mice don't develop the classical TDP43 pathology of motor neuron disease. So the question we asked now is whether riluzole may have an effect in mice that do have this pathology that's more reminiscent of what we see in people. Now, what we found is when we provided riluzole to the mice that have this MND pathology, we were unable to slow down the progression of the disease, and we're unable to affect the survival of these animals either. So this suggests that we need better drugs, clearly, and better drugs should have a better effect. So the question that we have now is, using these mice, can we identify drugs or drug targets that target this underlying TDP43 pathology that we know is the core pathology found in people with motor neuron disease? So in order to identify new drug targets, what we've been doing recently is performing large-scale protein profiling or proteomic studies in the brains and spinal cords of the TDP43 mice. So we've studied the brains and spinal cords from these mice before they develop any symptoms right throughout the disease course. We've profiled the levels of more than 6,000 different proteins at different stages throughout the disease in the brain and spinal cord of these mice. And we've identified over 150 proteins that are increased in levels even before the disease symptoms start in these animals. So even before there's any sign of disease, the mice still look normal, but we know that they will develop disease later on. We can see that if we look at the protein changes that are going on in the central nervous system, there's many, many changes already starting to happen. So when we delve closer and have a look at what these proteins are actually doing, we can find that there are changes over time as well in the levels of these potentially protective proteins. So I've shown an example here of a red protein, which we know is a protective protein. What we see in mice before disease onset, that there's not very much of this protein in normal mice, but before disease onset in these MND mice, they have really high levels of this protective protein. If we look later on in disease, though, we see that these MND mice return to the normal levels of this protective protein. So the question that we have from these studies is, if we're able to artificially drive this increased levels of these protective proteins throughout the whole disease course, whether that could be beneficial in disease. So one way that we're going about studying this is to use viruses for good. So developing new gene therapies in which we can artificially increase the levels of these protective proteins throughout the disease. So for these studies, we're using a small type of unharmful virus that normally is found in humans. They're called adeno-associated viruses. These are very, very tiny viruses. They're about 25 nanometers. So you'd need about 4,000 of them to reach the width of a human hair. They're very tiny. But we include in these the DNA instructions to allow the nerves within the mice to produce these protective proteins. So if we engineer these viruses and put them in the brains and spinal cords of these mice, it allows us to increase the levels of the protective proteins. And the studies that we're doing now are looking at whether increasing the levels of these proteins allows us to have a beneficial outcome on these mice. So with these gene therapies, they could be better and certainly more targeted with fewer side effects than classical drugs, which often have non-specific targets and cause changes that we're not really very able to tease apart. So the example that I mentioned that we're using these viruses for is to increase levels of these potentially protective proteins that we know are protective early on in disease but are lost later on. So as an overview of these studies, so we know that mice are a useful tool in which we can understand the early disease processes and also to test drugs. Recent work has allowed us to identify many new therapeutic targets. These are particularly related to the underlying pathology that's found in people with motor neuron disease. Our studies in the mice have shown that if we're able to target this underlying TDP43 pathology, then it does show benefits in the mice, which indicates that similar strategies in humans could be beneficial as well. And some ways that we could do that are using these new gene therapies or better drugs that are really targeted to that core underlying pathology, which we really hope will be beneficial for people with motor neuron disease. So I'd like to say thanks to the people in my lab at the Queensland Brain Institute at the University of Queensland. This is us out for lunch before COVID. All of our funding bodies, in particular M&D Research Australia. I was really happy to find out in the last few weeks that my lab's been successful in securing the Fat Rabbit M&D Research Grant from M&D Research Australia. And so we're really looking forward to taking those studies forward next year. So thank you very much, and I'm happy to take questions.
Okay, thank you, Samantha and Adam. They were fantastic summaries of your research and the field in general. So we've got a couple of questions that have come up. First one is from Monica Darcy for Sam. How long has this study been running, and how long till you publish the findings? I think that's talking about probably your second study you're talking about, testing the drugs.
Yeah, I mean, I can talk about both of them. The first study with the oligodendrocytes is written up as a manuscript, and we hope to submit soon, which is brilliant, implicating the role of oligodendrocytes in MND. The drug screening is certainly making progress, but it's sort of midway where we're still, I mean, we're testing so many drugs and on so many cell lines, but the team is working really hard. So hopefully we'll have more answers from that soon.
And there's a second kind of related question to that, which is a bit of a tough one maybe, but so is your research going to lead to a treatment in patients with an MND gene?
With an MND gene? Yes. I mean, the brilliant thing of the way that the lab is set up is that, you know, my interest from what I've shown is more in the understanding the cause of MND, looking at the pathways that are implicated. But we also have people in the lab working on drugs and drug screening. So I think the complementary nature of the scientists within the lab means that pathways we do identify, we then can translate to try and find drugs that target those pathways specifically. With regard to the stem cells, we are very fortunate to have many stem cells from patients with sporadic MND, which is important given its prevalence, but we also have many stem cells that have come from patients with familial MND as well, so with various genetic mutations. So we are trying to cover the whole spectrum of the disease as best we can.
We have another question here for Sam. There's some coming for you soon, Adam, don't worry. There's a question from David Lampard. Actually, he commends you both for thoroughly enjoyable presentations. And Sam, have you got any drugs that you've trialed that are looking pretty exciting? And then can you then share these with Adam for you to test in his mind as well?
Yes, I'll have drugs. Thanks, Sam.
Absolutely. And I mean, it's the brilliance of our MND community, and the funding allows us to collaborate and share our ideas. We certainly are finding some new drugs. I mean, at this point, we've got so many patient lines. We're testing the drugs that we know at this stage. So things like riluzole and other drugs that have shown some promise in similar neurodegenerative diseases. So we're setting up the pipeline to kind of find the phenotype and the efficacy, and then we'll screen the whole library of drugs. It may be further along than what I think, but I don't know any specifics that I can share right now. But Adam, I will let you know as soon as I know.
Great.
So Adam, this is a question for me actually. The question with the TDP43 mouse. So when you actually turn off the TDP43 clumping and get rid of those clumps, you said the mice basically get a bit better. Did you have motor neuron disease and motor neuron loss in those mice? And by turning off the clumping, do you actually get motor neurons coming back, regenerating, or do you just stop further loss of motor neurons?
Yeah, that's a great question. So we've done experiments in these mice where we've had up to 50% loss of the motor neurons, the alpha motor neurons from the lumbar spinal cord. So that's quite dramatic loss of motor neurons. And when we stop the continued accumulation of the TDP pathology, we see that we don't have any further loss of the motor neurons, but we don't have any new motor neurons forming either. So what happens is when we stop the TDP43 pathology, we stop any continued loss of the motor neurons. And what we assume is happening is that the motor neurons that are still there that haven't yet died are able to compensate and sprout and re-innovate the muscles, which allows for that functional recovery. I think there's probably a point where you've lost too many motor neurons and it's not possible for that mechanism to occur, but at least at about 50% loss of motor neurons, there is that possibility of the ones that are surviving to compensate for the loss of the other motor neurons.
So this is kind of a question for both of you. Is there any treatment yet that is showing that you can actually replace these motor neurons, either in like regenerating them in dishes or even in mice themselves? Has anyone been able to prove that there is potential to actually regenerate these cells?
Do you want me to take that one, Adam?
I'll let you take that one, Sam.
I mean, certainly, particularly the stem cell transplant experiments that I referenced, their goal is to replace the lost cells entirely with brand new cells. There are some deviations from that where we're trying to, some labs are trying to mimic the chemical secretions from these cells that promote the survival of the surrounding cells. There's drugs we can use that promote proliferation or increased generation of these cells, but they are all still preclinical. And I guess we need to keep in mind that, the homeostasis or the natural balance of the brain maintenance is very tightly regulated. And so, yes, we do want to increase these processes in M&D, but we need to find a way to do it that doesn't then go too far in the other direction. And that's certainly one of the risks of stem cell transplants, where we're going to cause other problems that we can't foresee. So it is really about finding ways to improve these pathways, but in a way that doesn't cause more issues, I guess.
Yeah, I think that's a good point, that I think it's also going to be hard to get a lot of motor neurons to be replaced in the spinal cord and then form the connections like they would normally do. I think that one thing from our studies in the mice may be that you may not need that, though. If you can support the motor neurons that are still alive to compensate, then that may be as beneficial as having some new motor neurons inserted in the spinal cord or the brain as well.
It was a much more optimistic answer than mine.
That brings us to some talks later in our series, because we'll be talking about biomarkers, and obviously a key to being able to rescue disease is going to be able to detect the disease early. We have a question here. Adam, can you make mice who have fast progressing MND versus slow progressing MND?
That's a good question. We haven't done yet, but we do have a project which is also funded by M&D Research Australia, actually, that we're about halfway through at the moment, where we're actually using viruses for bad. So I talked about in my talk using viruses for good, so in a therapeutic sense. But we are also developing viruses that we can use to specifically produce these TB43 proteins in normal mice to see if we can produce different behaviors and different forms of disease. So using those models, we'll be able to see if we have neurons that make more of the TDP43 protein, or or pathology that's been modified in some way, whether those mice develop faster disease, or whether we can find ways that we can have different types of TDP-43 models. I think it's important to study sort of slow and fast progressing diseases, but it is quite hard to model that in a mouse. It's often quite difficult to predict what you're going to get when you do try to produce a new mouse model of disease. So that is one of the difficult things.
I have a question here from Duncan Bailey. It's a stats question. So is the risk when you're screening lots of drugs that you're going to get a lot of false positives out of it, just from a sheer numbers game?
Oh, yeah, absolutely. It's certainly a risk. But that's why it's a much more comprehensive process than just screening the drugs once. So we do multiple wells treated with the same drugs, with the same cells. And so we can take an average of multiple wells that may counter for that variability. And then also, like I mentioned, we have a lot of patient lines that we're screening, so it'll allow us to make sure that the drug works efficiently on more than just the one patient motor neuron. Yeah, I guess that answers that question, I hope.
Okay, and this might be a very quick question, Adam. What happens if you get rid of TDP-43 completely?
That is bad. That is bad. Yeah. So TDP-43, it's an essential protein, which means that you have to have it in the cell. If you have too much, then the cell dies, and if you have too little, then the cell dies. So it's a hard protein to work with. But yeah, we can produce genetically modified mice that don't have any TDP-43, but they don't live. And also, if we increase the levels of this TDP protein, animals can't live either. So that is one problem with targeting this protein in disease, that we can't just sort of get rid of it completely, because the nerves are going to die if we do that as well. So we need to make sure that we decrease the pathology, but not get rid of the normal TDP-43 that has really important functions in the neuron as well. I guess that the hypothesis that we have is that TDP-43 normal function is important, so we need to maintain that, but in disease, for some reason, it becomes dysfunctional, it clumps together and has these sort of toxic effects that we want to stop without preventing the normal function from occurring.
And a question for Samantha. You mentioned Parkinson's as being a sort of example where stem cells of research has gone a bit further. Are there any examples where, in any other disease, not necessarily neurodegenerative diseases, where stem cells are now routinely, or at least have a successful example of being used to treat a disease?
That is a big question. Let me have a think. I mean, certainly stem cell transplantation is kind of a sexy area. A lot of the diseases are moving towards trying to bring it in. I'm trying to think of what I know. I do believe that there are some trials ongoing in developmental diseases, so diseases such as cerebral palsy, or things like lung disease associated with preterm birth, trying to transplant cells to try and increase the development that's been lost when they haven't been able to develop properly. So I believe that it has shown some promise in that area. I do think that we are still at the stage where it is being offered in some countries where the research hasn't quite caught up with what they're offering. And I think that's the case for many diseases. So in most of the countries working on transplantation, Australia is a big one, UK, America, we are trying to still nut out the preclinical aspects of it before it progresses too far, I guess, into the actual clinical trials.
Okay, just before we finish up, we had a couple of extra questions come in as well from on a slightly unrelated issue. So we have one is a question: does MND affect your bowels at all? I can answer that if you guys aren't quite sure. I think I can have a stab at that one.
I'll leave that to you, Gethin.
So I guess there's two aspects to this question. One is, does it actually affect your bowel function? And the answer there is not directly, but obviously with changes in your abdominal muscles and also changes in the amount you're moving and your diet, then this will have downstream effects on your your bowel function. A second sort of more research-orientated question is, can your bowel affect your MND? And there's a bit of research going on at the moment looking at your microbiome. So this is all the different types of bacteria that live in your gut. And there's quite a bit of evidence out there that the gut is actually linked to the brain. And there's a direct link between the gut and the brain. And that seems to be closely related to the makeup of your gut microbiome, all the different bacteria in your gut. So I know that a couple of our researchers we're funding, Derek Stein, who will be speaking in a week or two, as well as Pam McComb up in UQ, are looking at is there a relationship between your gut bacteria to either developing MND, or does MND affect your gut bacteria? And this is, I think, an area people are looking at in a lot of different diseases at the moment. And the thought is that this could actually be a route to provide a lot of information on a lot of different diseases. So watch this space at the moment on that question. The second question was just a general update on the status of copyright TSM and other advances in Australian clinical trials. Copyright TSM is at the moment finishing up its phase 2 trial. I know some of the patients have finished their trials, and we're still waiting for them to collate all the information and let us know to start publishing the details from those trials. There's a number of other trials going on in Australia at the moment. Tofersen is in the phase 3 trial for people with the SOD1 mutation. Again, this is in phase 3. It's coming to the end of it. We're hoping to get results early next year. There's the colloid gold trial is just commencing at the moment. We also have the Lighthouse 2 trial looking at the antiviral drugs. Again, this is about to commence. They've just putting all the pieces in place so they can start that trial. I think they're the main trials going on in Australia at the moment. Unfortunately, none are, no data is there at the moment, but we're hoping that early next year we'll start hearing about the results for some of these trials, and some of them will be getting into their phase 3 as well. So they'll be recruiting patients then as well for those trials. So, at the moment, it's just, I'm afraid, watch this space. So what I'd like to do now is obviously thank both of our presenters for doing a fantastic job of going through some fairly complex areas of research that are really diverse and sometimes can be quite confusing. So it's great to get that really good overview of those. This is part of a series. So next Tuesday, same time, same place, different Zoom link. Check your email links and your diary entries for when to come next week. And next week, we're going to hear about different studies looking at how we can improve care of our patients. If you have any more questions, please fire in on Facebook, and we'll be happy to answer those questions later, and just look in the Facebook link to see any more answers. For those people who logged in via Zoom, you'll be receiving an email with a recording. Probably takes about 24 hours for that to come through. And we'll also be uploading a recording of this session on Facebook as well, which you can watch at your leisure or send to your friends and colleagues as well. Again, that will probably take a day or so to get processed. So, and thank you for, as well as our presenters, thank you to the audience. Thank you for coming along. I hope you found this useful. And so good night, and please enjoy the rest of your week.
Thanks, Gaten.
Thank you very much.
Thank you, Adam and Sam.